Hello, I'm Jennifer Pearson with HESI. Welcome to our Roundtable, CiPA and Hindsight, Current and Evolving Roles of Human-Induced Pluripotent Stem Cell-Derived Cardiomyocytes in Drug Safety. I'd like to introduce you to our panelists. First, Dr. Gary Gintant with AbbVie.
Hello, my name is Gary Gintant. I've been interested in the safety of drugs, the cardiac safety of drugs, for my entire career, first starting in academics and then later on from an industry perspective. The use of these myocytes for early testing for safety, identifying safer drug candidates provides a unique opportunity for the industry to put the best compounds forward and to minimize hazard to patients. And it's a unique opportunity that needs to be addressed.
Thank you. Next, Dr. Alex Ribeiro with US FDA.
Hello, at the FDA, I am part of the Division of Applied Regulatory Science, and our mission is to bring new science to the drug regulatory process. And under this global mission, different folks in my division have been involved in the evaluation of iPS cardiomyocytes to be used in drug development.
Okay. And Dr. Jean-Pierre Valentin with UCB.
Hi, I'm Jean-Pierre Valentin. I'm a PhD scientist, and I've spent my entire career in the pharmaceutical industry in different companies, and more recently at UCB Biopharma. And working in the safety field, we are interested in the use of human-derived cardiomyocyte pluripotent stem cell and the application to the field of drug safety.
And Dr. Warong Liu with J&J.
My name is Warong Liu. I'm a scientific director at Janssen. I have more than 25 years on drug discovery and safety at Janssen, which actually we invest a few $1 million in last 5 years, use my cells to actually not only for human safety, also for 3R concept, which is reduce, replace, and refine animal use.
So as many of you know, the goal of the CiPA initiative is to develop in vitro assays that are more mechanistic for detecting proarrhythmic risk. And there are 4 main working groups: the clinical ECG group, the ion channel group, the in silico modeling group, and the myocyte group. The myocyte group is the one that HESI has been most involved with. And these are using the human-induced pluripotent stem cell-derived cardiomyocytes, or myocytes for short. We were working with the FDA using a grant called the BAA to fund some of the study here. And this validation study found a better than 80% concordance using the myocytes to detect proarrhythmic risk. So today we'll be talking about CiPA and what comes next. So Alex, can you comment on FDA's perspective generally on how the myocytes might be implemented in the cardiac safety process?
The specific technology of iPS cardiomyocytes is a relatively new technology. And at least in our division at the FDA, we look at them as an object of regulatory science studies. And the FDA in general has different priorities for regulatory science. And I see iPS cardiomyocytes as being potential tools to modernize toxicology. Also researching iPS cardiomyocytes because we can take them from different patients. We can actually look at different populations. So that's another priority, is to look at population-specific drug effects. And lastly, we're interested and we're looking forward to study iPS cardiomyocytes to be ready to evaluate the data that comes out of them once they start being used in INDs and applications.
And Jean-Pierre, how might these myocytes be implemented within the ICH guidelines?
Yes, I think, as you know, the ICH guidelines are ongoing, the discussions are ongoing, and will probably will further develop over the next few months and eventually a few years. I think it's important to recognize that the potential role of these human-derived pluripotent stem cells in such a context, and they may be used as part of the regulatory guidance, although this will be depending on the results that are still emerging as we speak.
So I'll ask our next questions to our pharma colleagues. So Gary, from your perspective in the pharma industry, how would you incorporate these myocytes into cardiac safety screening within your company? Are you using any assays now in early or late stage? And how many compounds would you say you're running each year approximately?
So Jennifer, pharma is always looking for the best compounds to move forward, both from an efficacy side as well as from a safety side. So I approach the problem from the safety side, and we're looking for answers regarding cardiac safety that are difficult to come by with animal studies. And these preparations, of course, provide us an opportunity to look at, let's say, human-relevant models for cardiac toxicity, and that can be of multiple forms. It can be electrophysiology, it can be functional contractility issues, it can be structural cardiotoxicity. So we're still learning because these preparations are still relatively new on the scene in terms of what their strengths and limitations are. We're learning what they are based upon comparisons of what we're finding in the lab and what others are finding in the lab as well, to what's been known about some drugs in the clinic. More the compounds or drugs have been successful, and those that have not been successful. So we're still evaluating these cells in a number of different assays, different platforms, to see which ones are more predictive and more useful for us in terms of defining the different forms of the cardiotoxicity. The numbers will vary depending upon the month or year, depending upon what we're finding in other tests that we're running, and what other companion molecules may have done, or in what things may have been seen in other therapeutic areas with the compounds we're working with. So right now it's on a case-by-case basis, and we're also watching what the industry does and what the regulatory guidelines are going to be regarding the use of these myocytes for screening as well as for regulatory purposes.
Great. So Warong, I'll ask you the same questions. And what's your perspective from the pharma industry on how these are being used? And are you currently using them? How do the data inform your projects?
Yeah, indeed. We actually invest a lot of money in the last few years. We validated the assay. Actually, we use as a screen platform, we screen 500 compounds per year, use this assay. And in future, we would like to upscale to do 1,500 compounds per year.
Great. So what is the perspective from the pharma industry? How do you incorporate these myocytes into the cardiac safety screening assessments? And how are you using this in early or late stage? And how is the data informing your projects?
There are human-derived cardiomyocytes are used in early stages of drug discovery in the hit identification in order to inform about which chemical series to progress, as well in the lead optimization in order to influence the structure-activity relationship. These different platforms are being considered as we speak, and we are working with different service providers to help us addressing the value of this assay. So far, it is anticipated that roughly 1% of the compound will be tested in this platform. That said, it is clear that there will be differences from project to project depending the specific needs of a given project.
Great. So Warong, you said that you've used these a lot. What is your experience with their predictive value?
So our experience based on the validation more than 100 reference compounds, the predictive value for lung QT is quite high, more than 90%. For sodium channel block, you can detect it, but you need to follow up more assay to do it.
Great. And Gary, what about you? What's your experience with the predictive value of these assays?
Yeah, so if I might, there was recently a study published. It was the HESI CiPA FDA validation study looking at the effects of 28 blinded compounds across 10 different sites across MEA and voltage sensing dye. We're using those both approaches, and we found very good area under the curve values for concordance between pro-arrhythmic risk as judged by the clinical effects of these 28 compounds with various predictors that were measured from the stem cell-derived cardiomyocytes in vitro. So we're quite enthusiastic about using these and bringing this data up to making it known to others, including regulators, in terms of understanding providing more a full integrated risk assessment based upon all the data that we have on these molecules.
Good. So Alex, so now that CiPA has introduced this paradigm shift towards more mechanistically driven acute cardiac safety assessment assays, can you explain your role and how these mechanistic assays might support that in our understanding with regards to CiPA?
I joined the FDA very recently, so the CiPA initiative was already on board and going, and papers that were already being published when I joined. So I joined late. My role in the field of iPS cardiomyocytes is to evaluate these cells. The effort of evaluation at FDA is being shared between different centers and different divisions. So, Center for Drug Evaluation and Research is where I am. Center for Devices also evaluating this technology, and CBER, Center for Biological Evaluation and Research, also evaluating. So there's a global concern also, and CTR, National Center Toxicology Research in Arkansas, they're also doing a lot of research. So there's a global concern on how these cells, this new technology, can impact drug development. To my knowledge, on the regulatory side, which I'm not very much involved, so far there are no INDs that have used data from these cells, but it's also my knowledge that data is always welcome in INDs to support what's being submitted. So
as we've learned, CiPA has helped validate these cardiomyocytes as a suitable in vitro model to address acute cardiac safety liabilities. They've already been integrated into routine testing at many pharmaceutical companies. They may soon be implemented into regulatory guidelines, but FDA has welcomed additional data in support of compounds. And we have seen that they do have good prediction to the clinic as well. So now, thinking about going beyond CiPA, the CiPA microscope has been rather limited. And so the HESI Cardiac Safety Committee convened a meeting back in May, and we wanted to look at another important topic, using these cardiomyocytes in a larger role to understanding various models of cardiotoxicity, especially related to structural toxicity assessment. So the cardiomyocytes may be related to stromal cells, have an opportunity to demonstrate a broader utility, and importantly, to fill a void where there's been essentially no predictive in vitro assays, and animal in vivo studies have been limited in predictive value for humans. So the HESI Cardiac Safety Committee meeting looked at something that we're calling failure modes. And these may be where we're failing to detect cardiotoxicities in our current models. So how can we be better at detecting these in a more mechanistic way? So we're hoping that we can address some of these using some in vitro strategies, and this is something that we will work towards addressing over the next few years. So now I'd like to take some time to ask questions of our panelists on how they're currently implementing in vitro structural cardiotoxicity assessments and biomarker studies. So Gary, can you tell us more about what's going on at AbbVie?
Yeah, so these cells provide a unique opportunity to assess structural toxicity early. And what I mean by that is to detect when drugs may be causing stress or injuring these myocytes before causing, let's say, the ultimate adverse effect, which is cell death. So we're looking for early signals where these cells are under stress, where these myocytes are maybe being injured. And part of, in terms of how we're doing this, well, we just looked to the clinic to see what's been done in the clinic to look for cardiac injury, for example, with heart failure or with myocardial infarction. There are a number of biomarkers out there that are used in the clinic, cardiac troponins, fatty acid binding proteins, some more exploratory microRNAs. These are pointing the way for us as to what should we be looking for in these stem cell-derived cardiomyocytes, and some very simple questions like, are they released? When are they released in terms of time course? At what concentrations of drugs, and how does it relate to the clinic, to what's been known about cardiotoxicity? There are some drugs, another thing I need to mention is that some drugs cause injury rather quickly, and some drugs cause injury over a long period of time. So we're talking about different time scales. With these myocytes, one can look at the responses of drugs, let's say a week, 2 weeks after continued exposure to the drug, to see what may be happening to the myocytes, which again is something that right now we can only look at with animal models, which again are not human models. So there are a number of opportunities here, a number of challenges that require some good science, some good investigators, both on the industry side, the regulatory side, and academic side, as to where these models are most helpful for detecting various forms of cardiotoxicity with different drugs.
Great. And while we're on, can you tell us more about what you're doing to assess cardiotoxicity?
Yeah, this is actually very interesting topic and challenge. So the early days we're missing because we're only doing acute assay, but in different projects we had in the past few years, we had kind of delay issues. So the cardiomyocytes actually offer us good opportunity. We can study in a delay assay, use different technology like impedance technology, even calcium transient, and then combine with gene expression and biomarker like Gary mentioned. So actually this helps us a lot to find a safe drug in the future.
Great. And Jean-Pierre, what about UCB? What are they doing to look at structural cardiotox?
Yes, first of all, it's worth reinforcing what my colleague said, that in effect there are three types of kind of cardiotoxicity. Typically the proarrhythmic events, and we talked about this earlier, then the cardiac injury and cardiac event, and we need to be able to address all these three components, and clearly the stem cell offer a great opportunity to address some of these. And to reinforce again some what my colleague said, there's two components, a temporal component, so being able through the stem cell to address not only acute but chronic effect, which is an important aspect. Second, being able to identify and develop and validate some biomarker, including some novel biomarker, and we have conducted some work on metabolomic, as well as some microRNAs that are quite promising biomarker of very early initiating event that would enable perhaps to translate this biomarker into an in vivo and eventually a human clinical situation. So a lot of promises focusing on this model using, again, as said by my colleague, different technology platform adapted to address different endpoints.
Great. And Alex, can you comment on FDA's research and how it might relate to assessing cardiac structural toxicities, and how maybe using stromal cells might be helpful to closely mirror the in vivo tissue composition?
Different FDA researchers have participated in meetings where all of these efforts have been shared and explained and elaborated. Personally, for me, what I say personally does not necessarily reflect the views of the FDA. I see a broad interest in looking at structural toxicity. I see a little bit of confusion on defining the structural toxicity, mainly the people that come from the in vivo side versus the people that come from the in vitro side. Normally, they may not be talking about the same thing. This is kind of the only caveat in the field that I see. Obviously, it's a new technology, as I've said before, and I see all my colleagues, including myself, we're very motivated to look at different modes of failure at the cellular level. The other question that's always comes up, and this is also another personal note, is the clinical or the physiological relevance of the measurements, which has already been raised. This is something that asking how to validate that with the currently available clinical data on drug effects is not a trivial thing to do. It's kind of just a personal comment. Yeah.
Thank you. So CiPA has provided a roadmap to validating this technology and approaches to using more mechanistic assays. However, the cells that are related to the stromal tissues may be a scalable test system to address this issue. So what sort of data do we think is necessary to validate a translational aspect using these stem cells to predict structural cardiotoxicity? So I'll open that up to the panel.
Perhaps I may start and stepping back a little bit. I think in order to get the confidence in an assay, there is probably three aspects that need to be covered. First is the confidence in the biology, ensuring that the biology of the test system mirror the biology of the species of interest. In this case, most of us are interested in human. Second aspect is the confidence in the assay itself, the robustness of the assay, and the intra and inter-laboratory differences or reproducibility. And finally, the third aspect is the confidence in the translation, which is typically achieved using reference compound or pharmacological tools, enabling to set up and define some parameters such as sensitivity, specificity, positive and negative predictive value of assay, as well as ROC curve. So I think we need to cover all three components in order to get sufficient confidence to use a given assay with our eyes open, understanding the strength as well as the limitation of these assays.
Something else, being an in vitro tool, a new in vitro tool. How this new in vitro tool compared to others? The availability of primary tissue, cardiac tissue, specifically human tissue, is not high. Even the ability to isolate animal primary tissue, it also requires a lot of training. It's not something that a lab can easily get trained on and acquire that capability easily. But I also see value in comparing the performance of this new cellular system under different situations, with stromal cells, without stromal cells, but comparing that to the state of the art or what has already been done since the 70s, since the 80s with primary tissue. There's high value in that also. In addition to doing the translational and the clinical comparison and comparing it to a clinical setting.
Yeah, the other value is, as Warong pointed out, you can do this without using animals. Yeah. And that's also a big advantage.
Yeah, I agree all the colleague comments, but for me, for the last part, I think important we have to find which kind of assay to measure functional parameter to compare the clinical incidence, and then second, to look the mechanism of action, because delay cardiotoxicity could be very broad, not like it just block hERG block.
So how do we see the integration of these technologies playing out, both from the individual company level and as it relates towards regulatory guidance and potentially inclusion in IND submissions? So maybe I'll start with you, Gary.
Well, that's an interesting and a difficult question. I don't think there's going to be any one answer. I think there are going to be a number of different platforms that come into play. And I think the trick to it, or not the trick, but as we get more data, we'll understand when to apply which assay, what the limitations are of each one, and use them to maybe gate the next ones. And again, taking an overall assessment from multiple assays, understanding where they best fit in the overall assessment of the drug as it moves forward, as well as maybe even looking for ways to prevent the adverse effects. I mean, if we're in a position where if we have an adverse effect, there may be a way to pharmacologically modify that effect with maybe another drug, and this may be down the road a little bit, but we can run clinical trials, so-called clinical trials in a dish, looking for drugs that may mitigate the adverse effects of single-agent therapies, and maybe keep these drugs alive and useful for patients farther down the road. It gives us a lot of flexibility, but we have to, again, understand the model and be smart about how we use the models.
Great. Alex, do you have thoughts on how this data could be useful for IND submissions?
It's my understanding, again, as I've noted before, that all data is welcome. All data that supports an application is welcome. To my knowledge, it hasn't happened yet, so there's no applications that use iPS cardiomyocytes. But I would like to completely support what Gary said. This first stage of evaluation was a very technology-based evaluation, which is extremely needed. So we had a new cell type that came to the scene. It enabled a lot of technologies that were not able to be used with another cell type. And I think right now we're at a stage where the next step, in my opinion, is mechanistic. And probably there, when we're looking at specific contexts of use for specific mechanisms of action, probably there it may be a good moment to start thinking, how can they be used to demonstrate efficacy? How can they be used to demonstrate a specific mechanism? Right now, at least in our division, something that we've been thinking a lot is, how can these cells be used to differentiate what is myocyte-specific drug effect from a non-myocyte-specific drug effect? It's kind of where we're thinking right now. And yeah, I think there's a lot still to come. I cannot give you a straight answer on how can they be used for INDs or for any application, either on efficacy or safety. But we're engaged in this evaluation, so we're looking forward for the right moment for that to come.
Thank you. So I've heard that these cells can be very useful for assessing both acute and possibly delayed effects. And there's a lot of promising biomarkers that could detect early events before the cardiotoxicity even occurs. However, we still really need to understand the confidence in the biology, the reproducibility of the assays, and of course the confidence in the translation. So HESI is committed to working towards this issue. We've recently submitted another grant to the FDA BA program. We're hoping to receive some funding in support of this effort. And if that happens, we will certainly be trying to further assess in vitro assays for cardiotoxicity. And so, we've heard a lot about how pharma is implementing this and how FDA has also been working on these issues and working towards long-term cardiotoxicity studies as well. And really that the myocyte technologies can improve the efficiency of preclinical drug development practices if we can work to understand some of their limitations. So most importantly, though, that these practices will result in safer drugs for people. So I'd really like to thank our panelists today. Thank you guys for your time and your efforts in the cardiac safety field. I really look forward to working on these issues further on our HESI Cardiac Safety Committee. And I'd also like to thank our sponsor, Ncardia.